Welcome, NEET aspirants. This question is from the chapter Biotechnology, Principles and Processes, and the question is on PCR. Following are the steps involved in the process of PCR. These are the steps. What you have to do is choose the correct sequence of steps of PCR from the options given below. So the first step is denaturation, is C. C is in option number 1 only. So this is the answer, confirmed. And mark it on the OMR sheet. 190 is 1. Okay. So this question is asked from one image. This is the one. So the first step is denaturation, then followed by annealing, and then treatment with Taq polymerase and deoxyribonucleotides. Then finally extension of the primer, and this is repeated 30 cycles to finally get the amplified DNA. C is denaturation, step number 1. A is annealing of the primers. Then D is treatment with Taq polymerase, third step. Then E is extension, step number 4. And finally, the result, end point is amplification 1 billion times. 3. That means 5. So step number 1 is denaturation. So what is the meaning of denaturation in the context of DNA? Denaturation simply means breaking of the hydrogen bonds. These bonds are broken. The hydrogen bonds which hold the two strands of a double-stranded DNA. And hydrogen bonds are broken with the help of high temperature. So 95 degrees Celsius temperature is maintained in the machine, the PCR machine. And then finally you will have the two strands separated out. So this will be 5' to 3' strand, and this is the anti-parallel complementary strand, 3' to 5'. Now that they have separated, next step will be primer annealing. The primer which is complementary to the sequence of our gene of interest, that will anneal. And this is called, so this base pairing of the primer with the target sequences of the gene of interest is called annealing. So usually the temperature is around 58 degrees Celsius. So this is important because the DNA polymerase cannot start from scratch. It needs a stepping stone. So primer provides the 3' end, and then DNA polymerase can add the new nucleotides according to the sequence complementarity and then extend the primer. Then step number 3 is treatment with Taq and dNTPs. So Taq polymerase is the DNA dependent DNA polymerase. It will associate at the 3' end. Source of this Taq polymerase is Thermus aquaticus. Now this is in vitro technique, and we need a polymerase which is thermostable. So Thermus aquaticus is a thermophilic bacteria. It not only survives, it thrives in high temperature. So this particular polymerase is isolated from this bacteria, and it has been useful, extremely useful in PCR technique. The next one is extension of the primer. So using the dNTPs, Taq polymerase will extend the 3' end. And then at the end, we will have two double-stranded DNAs. So the steps of denaturation, annealing, extension will now be repeated again. And this will be repeated 30 times. So if you do the maths, one cycle gives you two double-stranded DNA. So 30 cycles will give how many? So this x is equal to 2 to the power 30. If you calculate it, you will get around approximately 1 billion copies. Now this is an important point which I want to highlight here. If you go by what is given in NCERT textbook, your answer will be step number 1 is denaturation. Then step number 2 is annealing. Step number 3 is this, and 4 is this. Practically, when PCR is done, the first thing you do is prepare this cocktail, which is the master mix and then the primer and your template. This master mix will contain buffer and important ions like potassium and magnesium and ammonium to neutralize the negative charge on the DNA and primer. And then the tag polymerase is also there. Primer is used for the sequence specificity, and template is your gene of interest. So this cocktail is prepared and it is added in this Eppendorf tube. And then the lid is closed and it is put in the thermocycler, the machine. So thermocycler will have denaturation temperature, annealing temperature, and extension temperature all set in a program, and it will cyclically follow the program and to 30 cycles, and it will take around 1 hour, and after that amplification process will be complete. Okay, so if you go by the practical steps of PCR, the first step will become D. This becomes step number 1. And then denaturation is the second step. Then it will be annealing will be third, extension will be 4, and this will be 5. This is only for understanding. For NEET exam, stick to this one. Okay, because NEET follows NCERT textbook, and what is given in NCERT, that will become your perfect answer, the right answer. So always be a learner with an open mind. Okay? Try to understand under what context the question is asked. In this original question, D is given as step number 1, but then it is followed by amplification, and this is not possible. And therefore, we choose this as the correct answer of this question. That's all about this question. Thank you so much for watching my video.